In this video, I will talk about genetic variation in meiosis. In the previous video, we talked about the process of meiosis and the different stages involved in it. And we've got a number of different places where genetic variation occurs, which I mentioned at the time, but I'm going to talk about in a little bit more detail in this short video. So if we look in meiosis itself, we've got three places where we can have genetic variation. We have the crossovers that occur in prophase 1. We have got independent assortment in meiosis 1. And we've also got independent assortment in meiosis 2. We also, so in meiosis 2 as well. So that's in the process of meiosis. And another aspect we have, which is kind of part of meiosis, but also partly not meiosis, we have this idea of random fertilization, because we make the gametes during the meiosis, but then after the gametes are produced, any egg can be fertilized by any sperm. So it's another way we can massively increase the amount of genetic variation or genetic recombination. Crossing over occurs in prophase 1, so we can call them crossovers or we can call them chiasma if you're just talking about one of these things, singular, or chiasmata if you're talking about a number of these things. And what we get with these crossovers is they're in prophase 1 and we get an exchange of DNA between non-sister homologous chromatids. Horrible way to say. But what that means is if this is a homologous pair of chromosomes, so if we've got maternal in blue and paternal in red, remember the maternal and the paternal chromosomes are very similar in terms of the same length. They carry the same genes in the same position, but you will have different alleles of those genes. So if you look at the example we've got here, if we look at gene B, the maternal version has the capital letter B, and the paternal, the red version, has lowercase b version. And what you get with crossovers is we're going to get a sister chromatid from one homologous chromosome swapping with the sister chromatid of another homologous chromosome. And it's a reciprocal swap in the DNA, so it's an exchange of DNA, and what you end up with are combinations like this one down here. So what you can immediately see is that the blue chromosome now has a small bit of red on it, and the blue is now transferred to the red chromosome. But we've also got this swap in the DNA as well, because if you look at what we started with, we had the blue version had big C and big C, and the red version had little c and little c. And then because of where the cross occurs, you end up with one chromosome being big C, little c now, and the other one also being big C, little c. And we'll talk about this more when we talk about inheritance patterns, but this is a really important area of exchanging DNA, swapping DNA. And what we find with crossovers, it's not just one crossover in one location. So this is an example. This is taken by an electron microscope. This is showing 1, 2, 3, 4, 5 crossovers along the chromosome. So 5 locations where you've got this reciprocal swap. And some parts of a chromosome are more likely to have crossovers than others. Watch out for at university, but you call them hotspots. Hotspots are regions where this crossovers are more likely to occur. We also make new combinations of DNA in this idea of independent assortment, and we're looking first at independent assortment in metaphase 1. And what we end up with, we have the bivalents, so the homologous pairs of chromosomes that have paired up, and they line up in the equator of the cell, and they can line up in any direction. So you've got, in this case, the red going to the right, the black to the left, the red to the right, the black to the left. But this could happen in either way. I mean, it's independent of each other. So the black could go the other way, the red could go the other way, and the next chromosome pair is completely independent. So you get this independent assortment of the maternal and the paternal chromosomes after metaphase 1 when these chromosomes are separated. So just to show you the amount of variation you can get by independent assortment in metaphase 1, you can actually do a calculation for it. And the equation you're going to use is 2 to the n, where n is the number of chromosomes. And bear with me with my epic drawing skills. If we have a cell, there we go, that's a cell, so far so good, and in this cell the cell has one chromosome, so it's a very small, one chromosome only, and we are going to draw this one chromosome. So we have the red version and we have the blue version. And when we look at how these can be divided, there are only 2 combinations. So you either get the red chromosome in one nucleus or the blue in the other. So there's only 2 combinations. Helps if you spell the word combinations correct. So there's only 2 combinations. And 2 to the power 1 equals 2. Now, so that's one chromosome. I'm going to attempt to draw for 2 chromosomes. So wish me luck here. So 2 chromosomes. So remember, everyone has to be diploid. So we've got, should have drawn a bigger cell. So we've got maternal and paternal versions of everything. And if we look at the combinations we have, so we could have red and red, so one's longer than the other, and then we could have blue and blue, or we could have big red and little blue, or we could have big blue and little red. Oh, that's not red, is it? Or little red. Okay, so we've got 4 combinations, 4 combos. And then because we had 2 chromosomes, that's 2 to the power 2 equals 4. Now, I'm not going to draw 3 pairs, but what we've got down here is 2 to the power 3, which is 8 combinations. And hopefully you can see you could have all blues and all reds, or you have this combination, so on. Doesn't sound like a lot, but remember, humans, we have 23 pairs. So what we're looking at is 2 to the power 23. So just by independent assortment in metaphase 1, we've actually got 8,388,608 combos. I am not going to draw them out. So massive number of combinations. And this is not even accounting for the fact that during prophase 1 we've got crossovers, and that's not accounting for the independent assortment in metaphase 2. So massive and huge amounts of genetic variation, or what we call genetic recombination. During meiosis, we also have independent assortment in metaphase 2. So what we have here is the chromosomes line up at the equator. And when they line up at the equator, remember the sister chromatids are no longer identical. So they're no longer identical. So if we look at the combination here, we've got one sister chromatid that's all black, the other one that is black with a little bit of red. So they're no longer identical to each other. So it means that they're going to go this way, but they could go the other way. And the same is true for the other chromosome pair. So again, it is completely random which way they go, and again, it's a way to massively increase the amount of genetic variation there is. The final aspect to mention is kind of independent to the process of meiosis, but is kind of an outcome from meiosis. So we said meiosis is going to be used to make the gametes, the sperm and the egg. And what you get is the sperm plus the egg. They get fertilized. They're called a zygote, which is an early stage in development. And each gamete that has been made is unique. So we looked at why it's unique. So each sperm and egg is completely unique due to prophase 1 crossovers and the independent assortment in metaphase 1 and metaphase 2. So each gamete equals unique. And any male, so any egg can be fertilized by any sperm. And we've got massive numbers. So a female, this is the egg here, a female might release 1 to 2 eggs per month, roughly speaking. A male, each ejaculate from the male is 100 million unique sperm, and any one of those sperm has the ability to fertilize the egg. So massive amount of variation. So to summarize, we have looked at mitosis and meiosis in this topic, and we will be comparing and contrasting meiosis and mitosis in class together. But just to quickly mention some of the factors here, mitosis is 1 division. Mitosis is 2 divisions. Mitosis makes identical cells, what we call clones. Meiosis is different cells. We've got this genetic recombination. Mitosis is 2 cells produced. Meiosis, 4 cells are produced. Meiosis used to produce gametes that are haploid. Remember, we used that term N to describe it. Mitosis is used for body cells, and the cells are diploid, 2N. So that about summarizes the differences between meiosis and mitosis. We will talk more about inheritance in our next topic. Thank you.